Hello everyone, this is Dr. Corey Wisner, Associate Professor of Nutrition in the College of Health Solutions. And this week we'll be expanding on system science and pushing it more toward precision nutrition and really how to consider all potential variables that could be important in explaining the individual variability and responses to nutritional inputs. In order to discuss this topic, I think it's important to consider where we've come from in the field and the work that we're building upon. And so in this history, I think a lot of it goes back to the early 50s when James Watson and Francis Crick were able to completely identify the structure of DNA. And from that, we really expanded the field in the early 2000s to the Human Genome Project, where we were able to determine the entire sequence of the human genome, which is about 3 billion bases long. And sure, we thought that was going to sort of fix all of our problems if we just understood what genes primed different outcomes, that we would be able to improve health across multiple health-related fields. But of course that didn't work out perfectly, but this work was seminal to really expanding into other omics fields and understanding how socio-demographic sorts of information and behavior are also really critical for identifying proper treatments for people. So Francis Collins, previous director of the National Institutes of Health, really led the initiatives for not only the Human Genome Project, but also the Precision Medicine Initiative that the government has been running since 2015. And this all is built on the premise that after the Human Genome Project, we recognize that genetics explain an important but not complete portion of all of the individual variability in health. It's estimated that it's only actually about 30% of explaining the variability. And then, of course, there are other factors like health behaviors that are also really important, and we think those explain about 40% of outcomes. And then there are environmental factors, so where you live, what environmental exposures you are exposed to, and those are thought to explain about 30% as well. So after 2015, the scope of precision health has really expanded and started to acknowledge that other components, particularly behaviors, are also important. And so this led to more inclusive definitions of what precision health and, in the context of nutrition, precision nutrition really mean. But it all comes down to inter-individual variability and understanding why some people respond to treatments and others don't. So historically, nutrition research has always sort of focused in on a single nutrient, and that led to significant advancements, but we still haven't really fully understood the impact of diet on health. And we know that individuals all respond very different to single nutrients or combinations of nutrients, and that's really where we need to push the field to move toward. In addition to the response, there are a lot of things that can explain that response. So those differences could be genetically related, related to ethnic cultural practices, biological sex, how we regulate our food intake, so the timing at which we're eating the foods or how frequently. And there are a number of other psychological, physical, and biological factors that all come together to interact, and the timing of those interactions can also really impact what outcome you see. So the first approach to really kind of acknowledge that we can't just study a single nutrient and that many factors are interacting really in the nutrition field led to a two-sided way of explaining phenotypes for health, and those were nutrigenetics and nutrigenomics. Nutrigenetics is considering how your genes alter enzymatic function or hormonal activities in the body. And nutrigenomics is more about how the nutrients come in to then influence gene expression. So taking that into account, how do we create personalized nutrition approaches? Well, there's the U, which is blurred because there are so many factors that change our behaviors and what day-to-day life looks for us and what we're exposed to. And then at the most fundamental level, we have our cells, our DNA, that are also leading to the expression of proteins and signaling molecules that are also interacting with all of those external factors. And so at the end of the day, it comes down to, it's all about what makes our genetic code unique, but also how our behaviors and exposures interact with our genes to influence health. So as a fundamental place for us to start, we'll begin molecularly and just talk through the genetic components, since this is really where nutrigenetics, nutrigenomics began as a field. So genetic material is complex, but also very simplistic when you think about only having 4 bases. And so we have our DNA, which is made up of adenine, guanine, cytosine, and thymine, and those bases are twisted into a double helix that, of course, we don't want all of those genes to be regulated at once, so we have ways of tightly winding up and organizing the DNA so that it's not accessible for transcription. And that's where histones and nucleosomes come in, and then those bead-like structures that allow us to spool the DNA around them wrap up into a tightly packed chromosome. If we look within chromosomes, we have pairs from both mom and dad, so those alleles that you've learned about in biology, so the location of genes and whether it's a dominant, recessive, or heterogeneous pairing can change the expression or activity of different enzymes or proteins. And because of this is sort of the central dogma of genetics, where we have DNA that's transcribed into messenger RNA. That RNA leaves the nucleus and enters the cytoplasm, where translation occurs to turn it into proteins. We know that diet can alter the expression of genetic material, particularly because many dietary components are ligands for transcription factor and transcription receptors. We also know that we can metabolize various components and alter the concentrations of substrates and products or hormones that ultimately influence gene expression. That can also happen through signal transduction pathways, where we have G protein-coupled receptors that lead to internal signaling cascades, which then ultimately enter the nucleus for gene expression. At the genome level, single nucleotide polymorphisms, or just one base pair shift, can lead to very robust changes in protein activity because the structure then changes. So here you see where there's an AT match that's switched out for a CG match, and that could lead to a lower or non-functioning protein. And so there's variability among people where these single nucleotide polymorphisms ultimately create graded responses in how well our proteins and enzymes are able to act. There's also insertion and deletion polymorphisms, where instead of just swapping out a base pair, we're either adding a whole new one or deleting to shorten the base pair code. This can also alter the activity of proteins and enzymes. And then above the genome is the epigenetic layer of study, and this is really where we regulate how genetic sequences are expressed. And so we can do this by DNA methylation, histone methylation, adding acetyl groups or phosphates, and also how transcription factors are regulated. So this is happening above the genome, meaning that it's not related to what base pairs are expressed, but whether or not we're allowing those histones to unwind. And when they're unwound, are there still factors bound to the double helix that are blocking transcription factors from binding to express RNA? So DNA methylation is one way we do this. Not all genes get expressed, and one way that we silence genes is by adding little push pins or methyl groups to the DNA strand. So these help not only wrap DNA around histones, but also methyl groups can be bound to the DNA strand to block promoter regions from transcription factors. So we call that silencing genes. Many of these methyl groups actually come from dietary constituents. So folate, choline, methionine, and vitamin B12 are all intertwined in regulatory and cascade steps that basically pass methyl groups down through different nutrient pathways to ultimately enter the nucleus to regulate DNA. At the histone level, when DNA is wrapped around those little beads to keep it tightly compact, these little proteins are important for making sure that DNA isn't accessible in another way, and that it also isn't unwinding all over within the nucleus. This is really important for regulating DNA repair, cellular death, mitosis, and meiosis. And various molecular structures are used for this. So methyl groups, acyl groups, phosphates, ubiquitin, and even biotin, which is another B vitamin. Thinking about how dietary components influence gene expression, we can take a look at how polyunsaturated fatty acids are actually ligands for transcription factors like peroxisome proliferator activated receptors, or PPARs. So here you see the fatty acid entering the cell, interacting with PPAR alpha. That then enters the nucleus and dimerizes with another transcription factor, RXR, to ultimately allow transcription of this gene to occur. This particular sort of signaling cascade, or nuclear signaling, allows us to regulate fatty acid breakdown. It allows us to undergo beta oxidation of fatty acids to use them as fuel, and it also regulates our triglyceride levels and insulin sensitivity through the production of prostaglandins and leukotrienes. So next I want to just do a quick survey of a couple nutrition-related diseases and sort of where the field has come in understanding how different factors, particularly those that are within the genome realm, regulate our responses. And one of the most prominent and well-studied areas is really related to hypertension, and currently the national guidance for stopping hypertension or treating it with dietary approaches is through the DASH diet. So dietary approaches to stop hypertension is what DASH stands for. And this all leads to the fact that many people are salt sensitive because of the AGT gene. There are polymorphisms that happen that then affect the angiotensin gene that regulates sympathetic responses in the vasculature, blood loss, and leads to imbalances in sodium and potassium. So if you have an A polymorphism, that can make you more sensitive to sodium, so that your vasculature will constrict and lead to a higher blood pressure. And of course, you can have two A alleles that are dominant. You can have a recessive or a combination of both. And the expression of these polymorphisms varies across different demographics. So you'll see that amongst all people, it's about 66% of the population has these hypertensive related polymorphisms, and it's particularly prominent in African American populations, and you'll see that those of Caucasian descent have the lowest rate of expression of that polymorphism. Early studies that looked at individuals of European ancestry that were practicing different diets with less than 1.8 grams of sodium per day, or without sodium restriction at all for 3 years, looked at these 3 different polymorphisms, AA, AG, and GG, and right away you can see that the percentage of people experiencing salt sensitivity or hypertension is much higher in those not regulating their sodium intake. So as long as they have even a single A allele, they are having greater prevalence of hypertension. And if you reduce their sodium on the right side of this graph, you see that hypertension decreases. Other well-studied nutrient gene regulation pathways relate to the MTHFR gene. So this is methylene tetrahydrofolate reductase here, which leads to producing 5-methyl tetrahydrofolate, which is the only folate metabolite that can pass methyls further down the line through B12 and then methionine to produce SAM, or S-adenosylmethionine. This is where we basically are able to donate methyl groups to a variety of different things, particularly gene, the DNA within the nucleus. So lower intakes of folate have been associated with cancer, and many of these methylation patterns also have ties to cancer. The effects of low intakes are also magnified if someone is a high consumer of alcohol. And the polymorphisms specific to the MTHFR gene are, if you have the TT genotype, then you get, basically this protein here is less functional, and you get a buildup of 5,10-methylene tetrahydrofolate. So as long as you have a good folate intake, there are pathways that this can drive for cancer prevention. But if you have a low folate diet, then you're going to not only be methylating your DNA, but also have an increased cancer risk. And these pathways also have ties to the alcohol dehydrogenase polymorphism, which is where the link between cancer and alcohol consumption is coming in this case. And so if you have a polymorphism in the alcohol dehydrogenase protein, that's going to slow your ability to metabolize ethanol, and that then also increases your risk of colon cancer. Over time, caloric restriction has also become a well-studied intervention for improving longevity, and it's thought that this happens through the SIR2 pathway, which requires the B vitamin niacin, and basically it's helping to regulate how our chromatin is packed onto those little histones and nucleosomes and tightly wound into our chromosomes. It's thought that energy restriction increases the ratio of NAD to NADH so that this pathway can be better regulated. And the histone deacetylase is then activated so that chromatin stays tightly wound, rather than having the acyl group, which sort of unlocks it so that it can unwind. Next, I'll just briefly mention links between caffeine and breast cancer. So if someone expresses the BRCA1 or 2 gene mutations, their consumption of coffee may provide a greater decrease in cancer risk. And so this has been really interesting to see as a potential preventative for those who have this gene. And caffeine can actually influence estrogen metabolism, which is thought to be how it's linked to the BRCA genes. Other factors that we have to consider are copy number variants. So how many times you see a gene copy within a chromosome. And instead of having 2 copies, perhaps someone can have none, 3, maybe 30. And this creates significant differences among people. One example of a copy number variant that has shown a lot of links to downstream obesity risk is the salivary amylase protein. So the fewer copies that you have of this gene, the more likely you are to experience obesity and cardiometabolic disease. You can see that the genes in the map on the left also are tied to perhaps those who have long histories of eating higher starch diets. They tend to have more copies. So this is something that probably has evolved over time and is interesting to study in relation to interactions when then people change their dietary behaviors, move to different countries, and become more acculturated into the dietary practices in those new locations. And so at the end of the day, nutrients we've seen can act as transcription factors and regulators of gene expression. And an easy way to think about what their role is that a nutrient is a key, and it can unlock a receptor or regulate how that receptor or protein are acting. And so if we think about nutrients and receptors, many cells express little receptors on the outside membrane, and nutrients act as ligands to either cause signaling cascades to pass into the cell or for that nutrient to then internalize into the cell so that it can be used for different regulatory or metabolic pathways. And so essentially that nutrient is exciting the cell or activating it to do something. But of course it's much more complex than that. Various genes can cause lots of variability in the receptor's ability to act. Sometimes cells express a lot of a receptor, and so then those cells become hypersensitive to the nutrient when it's present. Others don't express as many receptors, so they might be hyposensitive, where there's more nutrient ligand than there is receptor to bind. And then lastly, there could be genetic mutations in that receptor, making it insensitive to the nutrient that's present. So changing gears a little bit, we can then look to different ways that people have tried to better understand these more nuanced responses that occur between nutrients and cell signaling and receptor expression. And this, I think, kind of began with the developmental origins of adult disease or early plasticity and priming. So this is basically saying that things that happen prenatally or very early in life basically alter your trajectory towards having or not having adult diseases like obesity, metabolic syndrome, diabetes, and cardiovascular diseases. And this is probably driven through methylation of genes, acetylation of histones, and possibly other epigenetic changes that are regulating expression, but not necessarily genetic defects or mutations in the genetic code. So this idea of developmental plasticity then says that the ability to tune our gene expression can create a phenotype that's best suited to a predicted environment. So in utero, if a baby is not getting enough nutrition, that is basically tuning their genes to say, hey, this environment is nutrient poor. When you're born, you need to be ready to thrive in an environment that's nutrient poor. So as long as that phenotype matches, that organism will remain healthy. But let's say that that nutrient poor environment in utero was actually driven by the fact that the placenta had some sort of issue and mom had eaten everything that she was supposed to, but the nutrients just aren't passing to the infant. Well, that's telling that baby, hey, you're going to live in a nutrient poor environment. But then when they're born, they don't have that placenta blocking them from nutrients anymore, and they're actually born into a very nutrient rich environment. And so that is going to lead to a phenotype mismatch and increase the susceptibility of metabolic disease. The first person who really explored this idea was David Barker, almost 2 decades ago, where he studied birth weight of babies who were born to women who lived through some of the European food famines. And they basically found an inverse relationship between birth weight and impaired glucose tolerance. So those who were born during that famine were poorly endowed with nutrition, but then the famine passed, and so then they're living in a more nutrient-rich environment, and so we know they were undernourished because their birth weight was low, but then they have a much higher chance of having impaired glucose tolerance as adults. This has also been observed for cardiovascular disease and obesity as well. A similar study in the U.S., the Rancho Bernardo study, had studied women born with low birth weights and looking at obesity and metabolic syndrome. So those babies born at a lower weight had a much higher prevalence of high BMI and metabolic syndrome later in life. And one of the best ways for us to study these generational impacts is to use animal models so that we can study how these influences of nutrient deprivation or poor nutrition influence multiple generations in relation to the risk for developing adult disease. So here, if we provide a energy-restricted low-protein diet in the maternal F0 generation, and then we have further breeding of multiple generations, and we can maintain energy restriction over time. And in the 3rd generation, if we want to then split to have energy restricted plus nutritionally adequate animals compared to controls who are nutritionally adequate all the time, we can then look at insulin sensitivity. And just by looking at the 2 graphs really quickly, you see on the top female panels of data, and then on the lower half males. There are different responses by generation amongst males and females, so there are sex responses in how insulin resistance persists over generations. And we can also see that over time, these generational impacts can wash out, where the insulin resistance in females, for example, is highest in the F2 generation, but then it comes back and improves in the 3rd generation, while still on the energy restricted, but even on the nutritionally adequate diet. So taking a moment to summarize what we've seen so far, I think we can summarize with 4 tenets of nutrient or nutrition gene interactions. First, we've seen that inadequate or excessive diets are important risk factors for disease. We've seen that dietary metabolites can influence our gene expression and alter the genomic structure, whether we're unwinding DNA or keeping it tightly compact. And the degree to which diet alters our health and disease risk really depends on individual genetic makeup, and that genes are affected by diet, those are likely to influence the onset, incidence, and progression of a disease. And so the basis of all of this led to, in 2023, the launch of precision nutrition research efforts and the All of Us program, adding in a robust nutrition portfolio of studies to really start to understand what diets are best for humans to have enhanced longevity and improved health long term. One thing that has really emerged with the launch of precision nutrition programming is better consideration of what's now considered the exposome. So specific external environment and genetic external environment interactions that all come together to alter the internal environment of our bodies and influence our risk for disease. And so some of those external environmental exposures are things like diet, physical activity, tobacco exposure, what's in the consumer products that we're exposed to. Whereas the external environment would be climate-related factors, pollution, social structures and structures of power that limit access to resources for certain populations. And when you start to think about all of those factors, this gets really complex. So many have tried to then take these ideas and define what we need for precision nutrition tools and methods, and there are plenty of different flavors to all of this. But I think generally people do consider and appreciate that we need to understand demographics. We need to have phenotypic measures like anthropometrics. Many people value the addition of mobile data, so tracking diet, tracking your physical activity. There are clinical and metabolic indicators that we can account for, and then of course trying to intertwine that with a lot of what we've been talking about is these genetic and omics type assays. A model that has been sort of thrown out there is that we basically need to have super deep phenotyping and consider a multi-omic integration to understand precise nutrition advice or come up with precise nutritional interventions for people. But if you look across this, it's then, it has medical and family history, but it's not really getting at any of the, or socio-demographic factors, I guess I should say, that we need to also consider. One model that has done, I think, a decent job of trying to bring in all these vast factors that are often studied in isolation is the life course health development model. And it really tries to take into account environment, biology, behavior, developmental adaptations, and also social experiences, so early trauma that could impact downstream health outcomes, and really looks at this as sort of a trajectory model, and that there are inflection points where certain things could be intervened on for improvement. And so these sorts of frameworks will, I think, be really important for us as we design projects and think about what factors we need to be measuring in our studies. And so that brings us to our last slide of what do we really need for this field? Well, we need to understand nutrition, metabolism, and health, which many of you who are coming from nutrition backgrounds are already spent considerable time studying. We need to understand the phenotypes of individuals. What are the outcomes that they're experiencing or at risk for? With regard to nutrition specifically, we need validated nutritional intake measures. And right now, we don't have the greatest measures. And so we're kind of having to do the best with what we have. And ultimately, what we want to do is come together and merge all of this with omics so that we can personalize health so that people respond appropriately to the treatments that we prescribe. And this stuff will then further lead into policy where we have then health claims for functional foods and other ways that this can be doled out to the general public. And so across all of this, we have mechanisms, biomarkers, better assessment techniques, personalized nutrition, understanding what's right for that person culturally, as well as genetically, and then at a health claim or policy level, we need to roll out society-wide methods of implementing all of this so that it's fair and equitable for all. And that, I think, leaves a lot for us to do, to work on, and many of you are all working in, different areas of this space, but it's important to consider how all of this really can come together, and how when we work in teams, we will be considering all these different pieces of the puzzle, and that's going to move science forward so much faster. Thank you for your time, and if you have any questions, feel free to reach out to me. And just remember, this is just a little primer to all of this. This is far more complex, and there's lots more that we can cover, but I hope that this helps at least give you some perspective on where we've come from and where we're headed for precision nutrition.